Now let's talk about the neuromuscular junction. So the skeletal muscle needs a signal from the brain for contraction because these are voluntary muscles. Somatic motor neurons are the nerve cells that connect to the muscles, and the nerve cells of somatic motor neurons are in the brainstem and spinal cord that serve the skeletal muscles. Now, the somatic motor fibers have their axons that lead to the skeletal muscle, and that's how they help in contraction. Now, the axon of a motor neuron are called somatic motor fibers. Each muscle fiber is supplied only by a single motor neuron, but each neuron may branch out and serve multiple muscle fibers. Now, a motor unit is one nerve fiber and all the muscle fibers innervated by it. And usually a single motor unit contract in unison, so they contract together. And if for effective contraction, usually it requires the contraction of multiple motor units all together. And we have an average of 200 muscle fibers for each motor unit. And for example, an eye muscle requires contraction of 3 to 6 motor fibers per motor unit. So the eye muscle motor unit over here is a small motor unit that provides a very fine degree of control. For example, eye muscles and hand muscles. And then gastrocnemius are the muscles of the calf, which have over 1,000 muscle fibers per neuron. So these are large motor units and they provide more strength than control. So the neuromuscular junction contain synapse. Now this is the point where a muscle fiber meets the target cell. When the target cell is a muscle fiber that synapse, it's known as a neuromuscular junction. Now, in the process of muscle contraction, chemical messengers called neurotransmitters are released into the neuromuscular junction and provide a signal to allow muscle contraction. Now, at the point where the synapses meet a muscle fiber, there is a cleft that separates the muscle fiber from the axon. And this is called the synaptic cleft, or the opening where the neurotransmitters are released. So the axon terminal contains synaptic vesicles that are filled with neurotransmitter, and when we think about moving our hand, the signal is sent from the brain along the sensory motor neurons that contain synaptic vesicles filled with acetylcholine. The electric signal causes exocytosis of the vesicle, and acetylcholine is released into the synaptic cleft. And at the neuromuscular junction, the muscle fiber has acetylcholine receptors that respond to the release of acetylcholine. This picture shows an artist rendition of a neuromuscular junction. So right over here is the synaptic knob, which is a swollen end of the nerve fiber. The synaptic knob contains synaptic vesicles filled with acetylcholine. And when signal is sent for muscle contraction, these acetylcholine molecules release acetylcholine in the synaptic cleft by exocytosis. Now, right around the synaptic cleft, you see this specialized cell known as Schwann cell. So Schwann cells basically play a very important role in insulating the axon. So they form the myelin sheath around the peripheral axons of the peripheral nervous system. And myelin is a fatty layer that actually provides insulation to the axons in the peripheral nervous system. This picture over here shows the neuromuscular junction, the microscopic image of the neuromuscular junction. And right over here is a muscle fiber, and this is the motor end plate of the axon. And all these nerve fibers are the axon of a motor nerve that brings signal to several different muscle fibers. So the muscle contraction begins at a giant motor neuron, and this neuron conducts an impulse down the length of an axon. And an electric impulse is in general a special self-propagating electric signal that can only be initiated by special structures like the axons of the neurons. So electrically excitable cells help in muscle contraction, and both muscle fibers and neurons are electrically excitable because their membranes exhibit voltage changes in response to a stimulus. And this study of changes in the electrical activity of the cell is called electrophysiology. But muscle contraction and relaxation occurs in 4 major stages. The first stage is known as excitation. Excitation is a process in which a nerve action potential leads to a muscle action potential. So a nerve sends an action potential down the axon, and this in turn leads to a change in the action potential or depolarization inside a muscle fiber that creates an action potential in the muscle fiber. The second stage is excitation contract coupling. Now, excitation contract coupling is the linking of action potential on the sarcolemma to activate myofilaments. Now, this is the event that prepares the myofilaments for contraction. Now, keep in mind the muscle is full of proteins, and these proteins have active sites. And when something when something needs to be activated, it usually involves change in the structure of protein molecule. Now, the next stage is contraction. Contraction is the contraction of muscle fiber, and during contraction, tension develops in the muscle fiber, and the muscle fiber may shorten. But again, this depends on several factors, but in general, the contraction and relaxation of the muscle fiber is called the sliding filament theory. Now, the last stage of muscle contraction and relaxation is obviously the relaxation. Once a muscle contracts, it must relax and go back to its normal form. So when stimulation ends, a muscle fiber relaxes and returns to its resting strength. So the first step in muscle contraction is excitation. So when the nerve signal arrives at the synaptic knob, the voltage-gated calcium channels open and calcium enters the synaptic knob. This stimulates the release of acetylcholine into the synaptic cleft, and this happens by exocytosis. The synaptic cleft now has acetylcholine released into the synaptic cleft. So one thing we briefly talked about when we were looking at membrane transport was the resting membrane potential. So the resting membrane potential, I need to write this down. The resting membrane potential of a cell is the charge difference or voltage difference across the cell membrane when the cell is at rest. So the resting membrane potential of a muscle cell is minus 90 millivolt. Okay, so when the acetylcholine receptors are released into the synaptic cleft, the receptors on the muscle cell that are ligand-gated sodium channels, they get activated. So this takes attachment of two acetylcholine molecules to each receptor, and this receptor is a ligand-gated channel. So two molecules of acetylcholine bind to the ligand-gated channel, which causes this channel to open. And these channels are sodium channels, so when sodium-potassium pumps. So when these channels open, sodium enters the cell, the muscle cell, and this rush of sodium into the cell causes the membrane potential to shift from minus 90 to plus 75 millimole. Then potassium exits the cell and the membrane potential returns to minus 90 millivolt. So this quick change or shift in membrane potential is known as end plate potential. So this quick change in the shift in voltage, which is known as end plate potential, this causes nearby voltage gated channels to open up, and voltage change in the end plate potential opens up the voltage gated channels, which further produces an action potential. So more sodium rushes into the cell, which causes the cell to depolarize, and this creates an action potential, which is sent down the sarcoplasmic reticulum. So when the action potential is strong enough, it leads to the next step known as excitation, contraction, coupling. And the action potential spreads down the T tubules. And the T tubules, if you remember, T tubules are present at the ends of the sarcoplasmic reticulum, sandwiched between terminal cisternae. So the action potential spreads down the T tubules, and this opens up voltage-gated calcium channels. And calcium leaves the sarcoplasmic reticulum and enters the cytoplasm of the cell. Okay, so this action potential that was sent down the nerve terminal causes release of acetylcholine. Acetylcholine receptors bind to the ligand-gated sodium-potassium channels, which causes an end plate potential. The quick shift or the end plate potential in turn leads to the activation of voltage-gated potassium channels. Now, when so much voltage-gated sodium channels, when so much sodium enters the cell, it causes an action potential, and the action potential is transferred down the T tubules, which in turn causes the release of calcium ions into the cytosol. So this is where all the action happens. We need the release of calcium ions to cause muscle contraction. So when calcium binds to when calcium binds to the troponin complex over here from the cytosol, troponin changes shape and pulls on the tropomyosin molecule. Now, troponin loves calcium ions. So if there's a lot of calcium ions in the cytosol, troponin is going to attach to calcium and change shape. This causes troponin to pull upon the tropomyosin molecule, which causes the troponin-tropomyosin complex to release the active sites on the actin molecule. Now, the myosin head then binds to the exposed actin molecule and forms a cross bridge. So this picture over here shows a cross bridge formation between the myosin and actin filament, and this is an energy consuming process, so it requires the presence of ATP. So the first step that happens for myosin to get ready for attachment is the hydrolysis of ATP to ADP and PI. So this ADP molecule activates the caulking or docking of the myosin head to the actin molecule. So you can see that in this picture, the myosin is in a stretched out state and it has an ADP and PI attached to it. So next what happens is the formation of a cross bridge when actin sites are exposed. And during this time, the myosin molecule releases the ADP and PI, and the energy released is used to pull the actin inward towards the sarcomere. Okay, and ADP and PI are released, and this release basically causes the formation of ATP back from ADP and PI molecule. So this free phosphate combines with ADP to forms ATP, which can be reused for another contraction cycle. So when myosin releases ADP and PI, it pulls the thin filament towards the sarcomere, and this is called a power stroke. And upon binding more ATP, myosin releases actin. So when ATP is formed from the free ADP and PI, this ATP binds again to the myosin head, and the process can be repeated, meaning the myosin gets stretched again and it binds again to the active site of actin. So in this way, each head can perform up to 5 power strokes per second, and it's an energy consuming process. So the last step is relaxation. A relaxation process ultimately begins with the motor neuron, when the motor neuron stops releasing acetylcholine into the synaptic cleft. Now the muscle fiber repolarizes, which closes the sarcoplasmic reticulum gates. Now this causes all the excess calcium in the cytosol to be pulled back into the sarcoplasmic reticulum. And the calcium ions from the troponin molecules get released. And when the troponin molecule loses the calcium ions, it goes back into its normal state and it changes conformation again, thereby closing the active sites of the actin molecule. Now, this picture just shows calcium being pumped back into the sarcoplasmic reticulum by the process of active transport. Okay, as we discussed before, relaxation is caused by removal of calcium from the troponin molecule. So the troponin complex basically reblocks the active sites of actin, and when the troponin sites are reblocked, myosin can no longer attach to the active sites of actin. So I will be posting an animation of muscle contraction that will help you understand the process better. So please watch the video, which is being posted right next to this video.